I am going to walk you through the cell cycle.
So this is the cell we're going to be working with today. So this is the cell membrane, the nuclear envelope, the DNA, and the centrioles. So interphase is basically just an extended period between cells. So cells are actually in interphase the longest. So the first part of interphase is actually gap 1, which is also known as G1, and this is when the cell is going to grow. So as you can see, the cell membrane got bigger, and then the proteins that are necessary for the division of the cell are actually going to be made in here as well. So then the cell goes through a checkpoint. So the checkpoint is basically just making sure that the cell is large enough, has sufficient nutrients, and then it has enough growth factors present. So if the cell either doesn't make it to the checkpoint, it can also go through G0, which is a stable stage. So it's a non-dividing phase, which is basically just like a waiting period to go to the checkpoint in order to get into the synthesis phase. So the waiting period is due to regulatory signals. So the next phase would be the synthesis phase, also known as S phase, and this is when the DNA is going to be in its uncondensed form, as you can see now, and then it's actually going to be duplicated, so there's going to be 2 sister chromatids. These are the 2 chromosomes, and then when they are duplicated, it'll be attaching by the centromere, which is right here, and then these are going to be the 2 sister chromatids. So still in its uncondensed form, it's going to enter the gap 2 phase, which is G2. Basically, there's going to be more biochemical reactions, and it's going to be preparing for division. So it's basically to check and see if they have if the cell has all its components, and that the DNA is duplicated. And then there might be a little more growth in this stage. So the next part of the cell cycle would be mitosis, which mitosis is basically just the separation of the sister chromatids. So the first phase of mitosis is prophase. This is when the duplicated DNA is actually going to condense into chromosomes, and they're going to do this using histones, which is a protein. So also in the prophase, these spindle fibers, which are these white things, or also known as the microtubules, are going to form. And then the reason that the DNA condenses is it can help it move about without being tangled within the cell. So the next phase would be the prometaphase. And the first part of this is basically the nuclear envelope is going to start to disintegrate. And since this doesn't break apart, we're going to pretend it just disintegrates. Okay, and then the spindle fibers are actually going to attach to each chromatid, to each chromatid's kinetochore region from each pole. So this is what the cell would look like when the spindle fibers are actually attaching to the chromatid's kinetochore. And then this is the centrosomes at each pole, this right here and this one over here. So the next phase is going to be metaphase. So this is when the chromosomes line up on the metaphase plate, which is just the middle. So this would be the middle of the cell. And then they occasionally overlap, but since obviously there's not that many chromosomes in this cell, then they don't really have the need to overlap. But when you have multiple chromosomes, 40 something or how many ever, they usually have to stack up almost. So after metaphase, there's actually a checkpoint, and it's basically looking to see if the spindle fibers are actually attached and that the chromosomes are lined up. So in this cell, it looks like the spindle fibers are attached and that it's lined up in the middle of the cell. So the next phase is going to be anaphase, and this is when the sister chromatids get separated from each other and they move towards the opposite poles. So as you can see, these 2 yellow ones were together, and now they have moved towards the opposite poles, and these 2 green ones were together and they have moved towards the opposite poles. So the reason this happens is because the spindle fibers, which are the white pieces, they are actually getting shorter, which is drawing the chromatids to each pole. So a lot of things happen next, and the next phase is telophase. So this is when the nuclear envelopes are going to actually reform around each set of chromosomes. And in the meantime, the chromosomes are actually going to uncondense. As you can see, there's green and yellow, both uncondensed in both nuclear envelopes. And then the cells are actually going to start to pinch off, which is where it's starting to divide, and eventually it'll start to close in and pinch off for separation. So as you can see that through telophase that the cells have started to pinch off, So the next phase would basically be cytokinesis, and this is the cells actually separating. So now we have two different cells. And cytokinesis is basically the cytoplasm divides and the two daughter cells actually split completely. So both cells are actually going to be identical to each other and the parent cells. And that is the life cycle of a cell.